We are going to move on to our next presentation by Dr. Roxana Myers. It is entitled Management of Plant Parasitic Nematodes in Ornamental Crops. Dr. Myers received a Bachelor of Science degree in Tropical Horticulture from the University of Hawaii at Hilo and a PhD in Plant Pathology specializing in Nematology from the University of Hawaii at Manoa, College of Tropical Agriculture and Human Resources. She is currently employed as a research nematologist at the USDA's Daniel K. Inouye U.S. Pacific Basin Agricultural Research Center, where her responsibilities focus on the development of cost-effective and sustainable approaches to the control of plant parasitic nematodes. Her areas of research include evaluating methods of media disinfestation, testing the efficacy of nematicides in Hawaii's agricultural systems, utilizing predatory nematodes and entomopathogenic nematodes as biological control agents, and developing crop plants with resistance or tolerance to plant parasitic nematodes. Dr. Myers, thank you for joining us today.
Thank you very much for the great introduction, and thank you to HFNA for asking me to present my research today, and also for this beautiful lei.
You're welcome.
So the focus of my talk today will be management of plant parasitic nematodes in ornamental production. And as Glenn mentioned, I work at the USDA Pacific Basin Agricultural Research Center, and this is a photo of our beautiful facility in Hilo, Hawaii. So plant parasitic nematodes, just a quick review for any new viewers that we have this year, are microscopic non-segmented worms. The stylet mouth part is for piercing the plant cells, and they cause billions of dollars of damage to agricultural crops worldwide. Today we'll be focusing on burrowing nematode, Rhodophyllus similis. It's a migratory endoparasite which is found in the roots and gobo of Anthurium and is commonly disseminated through infested cuttings. So how do nematodes damage crops? Well, they destroy and weaken the root system, which interferes with the uptake of water and nutrients. They cause stunting of the plants and poor crop yields. And in Anthurium, they reduce the number of flowers and the size of flowers. And this photo is of Anthurium roots infested with burrowing nematode. Kind of at the middle stage, you can see the necrotic spots, lesions on the roots, and then eventually that turns into kind of that brown root rotting, and secondary pathogens are able to come in through the wounds caused by the nematodes. But don't worry, if you have them, we have a solution. I'm gonna explain to you an integrated pest management plan for controlling these pests. I'm going to break it down into 4 components: prevention, eradication, host resistance, and curative control. First, let's start with prevention. If you don't have them, you don't want to get them. So start clean and stay clean. Start with clean propagative material and volcanic cinder media. Watch your water movement from surrounding beds and fields. Train your staff in good sanitation practices. Start work in nematode-free areas first, and then moving to infested areas last. And remember to disinfect your tools and equipment, especially when moving from infested to non-infested areas. Okay, second step, eradication. You need to disinfect your propagative material. If you're using gobo cuttings, it's very important that you disinfect them with a hot water bath, because even symptomless cuttings can have high nematode populations inside. So we recommend a hot water bath of 50 degrees Celsius for 15 minutes to kill nematodes. And if you're worried about blight, you can extend that to 24 minutes. As you can see here, there's 2 systems, a small and a larger system on the bottom. But you notice there's 2 tanks, because one tank is for heating the plants and the other tank is for cooling them down. So it's crucial that you immediately cool the plant material down to room temperature, or it's going to continue to cook and you're going to see heat damage to the material. Okay, some growers have incorporated this technique with good success. They start by heat treating the gobos and laying them in trays. And then you can see how nicely the material came out as it grew out. Some cultivars are more tolerant to heat than others. So I would recommend taking this slow and just doing a few of each cultivar first. And then you'll get a hang of which ones can handle the heat better than others. And definitely on your first run, don't do a giant batch. Just take your time until you get comfortable with the technique. So the next step for disinfestation is using steam sterilization of your cinder media. You can do that by steaming in a soil cart on your own farm, or you can purchase steam cinder from Sanford's, who steam it directly in the back of their truck and will deliver it hot and fresh to your farm. So the temperature needs to get up to 100 C, which is 212 degrees Fahrenheit, for 30 minutes at the center of the media. And if you're using a soil cart, this will take approximately 2.5 hours. You can also do pre-plant steam disinfestation directly into your cinder beds. We use a rubber mat which forces the steam down into the beds. And it's the same idea. You need to get up to 100 degrees Celsius or 212 Fahrenheit for 30 minutes. And this usually takes approximately 2 to 2.5 hours per row. If you use this technique properly, you'll get complete eradication of plant parasitic nematodes. This is a photo of some of the other types of equipment. So you can get a much smaller generator system or a much larger, fancier system. There's quite a variety to choose from. This technique is used by a variety of growers. This is the bottom left photo is of a flower grower in Volcano who's steaming directly in his greenhouse beds in soil. And on the right, we removed compost media from greenhouses and we're steaming them on concrete slabs and then returning the media inside. Because they were doing trellises and growing vegetables, we couldn't lay the mat down flat inside their greenhouses. But there's always a way. If you're interested in learning more about this technology, please contact me. Alternatively, you can disinfest the cinder beds with Vapam. Although this is a restricted use chemical and has a little bit, is more restrictive and has more requirements into its use. But ideally, if you have enough funding to go this route, you can do Vapam fumigation of the cinder beds and then start with nematode-free tissue culture plant material. And this is your ideal situation to get started early on. We start clean and stay clean from here. Okay, let's move to host plant resistance. The benefits of host plant resistance are the reduced use of nematicides, reduced cost of control, and no change to normal farming practices. So host plant resistance can be developed in a number of ways. One is through conventional breeding. You look for germplasm with resistance or tolerance to plant parasitic nematodes. A lot of work has not been done. This is an area that needs exploration in Anthurium, but we know just through some of the trials that some cultivars are definitely more tolerant to nematode infestation than others. The next thing you can do, which is not really an option for Anthurium, but in other crops like coffee, is grafting with nematode tolerant or resistant rootstocks. And the third way to develop host plant resistance is through genetic engineering. You can insert proteinase inhibitors, which interfere with the nematode's digestive system, or you can do a technique called RNAi for gene silencing of important genes of the nematode that cause parasitism or potentially reproduction. And just on that note, healthy plants are more tolerant to nematode infestation. So with optimal growing conditions, plants are better equipped to fight off nematodes. So if you optimize your fertilization and soil health and control your insect pests and diseases, your plants will be stronger and healthier and able to withstand this type of pathogen. So if you do get nematodes, eradication doesn't work, prevention, eventually the nematodes move in, there is an option for curative control, which I'll discuss now. So we've been working on a number of nematicide field trials. We've done 6 one-year trials in the past 6 years, and 4 of those we've been focusing on the chemical fluopyram because that seems to have the most success. So this is just a quick overview photo. The fluopyram treated on the left side, you can see is much healthier than the untreated controls, including the foliage and the root system and the size of the flowers. So through all of the last 4 studies we've done, we've seen that nematode populations were reduced by fluopyram applications. There was robust root development and improved plant vigor in treated plots, and an increase in flower size and yield, which is, of course, that's what the growers want to see. So we're very excited about that, and we do think that it has potential in mitigating the damage caused by burrowing nematode in Anthurium cut flower production. So there's many formulations available. Currently, the only formulation available for ornamentals is called Broadform, which is a fungicide that contains trifloxystrobin. But in addition to that, it also contains the same amount of fluopyram as the active ingredient. And which is nice about this product is that most likely the fungicide will help to maybe mitigate some of those the secondary pathogens that are coming in and causing that root rot that we see in the Anthurium roots. So just a quick overview of some of the results we got previously. We see the number of plant parasitic nematodes was reduced in Luna, which is the same thing as Broadform, and somewhat in Indemnify, which is a product just containing the fluopyram. So the plant vigor was much increased with the application of the 2 fluopyram formulations. And most importantly, the flower harvest was improved, not only the quantity of the flowers, but also the size. So the recommendations for fluopyram use are to apply at the first sign of a burrowing nematode infestation. Target the application to the root zone under the leaf canopy using drop nozzles. And plants with, what we learned was plants with severe damage from nematode infestation won't recover as fast with chemical treatment. So it is important, as soon as you think that nematodes did come into the field, that you try to treat that and address that problem right away. Because when your plants are damaged, it takes much longer for the new root growth to occur and the plant vigor to come back. So during that time, you're losing production, so get on it as quickly as possible. So now I'd like to talk about a couple of the research projects we did this year, just to give you an update. I'll start with the most recent fluopyram experiment. And this is done in cooperation with the University of Hawaii at Manoa, at Komohana, and at the Manoa facility with Brian Bush, Joanne Lichty, Dr. Arnold Hara, and Dr. Brent Sipes. So this last year, we did a nematicide box trial at Waikea, and this was the first time we didn't actually do the experiment on a grower cooperator farm. And the reason for that was I wanted to do a total crop destruct and be able to pull up all of the plants and evaluate their root systems by root health and root weight, and then maybe get a more definitive number of the populations inside the nematode, the population of nematodes inside the plant. But this cultivar, as you can see, a Toyama peach, very beautiful and pretty tolerant to the nematodes. So we didn't see as much differences as we would have with a more susceptible cultivar. So for this nematicide box trial, we wanted to work some more with the fluopyram, but also evaluate some of the new nematicides that have come on the market. This is a new time for nematicides. For a long time, we didn't have anything new. We lost the use of Nemacure, and there was kind of a time where there was no options for chemical management. But this is kind of a new era where these nematicides are less environmentally harmful, and not on the restricted chemical list. So open for more use by farmers. So the ones we tested were broadform, which is also labeled as Luna Sensation, which contains the fluopyram and the trifloxystrobin. Also vellum and indemnify, which is also indemnify, were tested. And the new chemicals we tested were Nimitz, whose active ingredient is fluensulfone, and Venerate and Magistine, which is a Burkholderia species. And we compared those with an untreated control. So as I mentioned, we use the cultivar Toyama Peach, and thank you very much to our grower collaborators who provided the plant material for us. We inoculated these cinder boxes with burrowing nematodes, 2,000 per plant, and we had 10 plants each in each box and 5 replicates per box for treatment. We did nematode sampling of the cinder and roots quarterly, and we're currently working on final whole plant assays. Weekly flower harvest and sizing was also conducted. So one thing I noticed right off was, even though this cultivar seems to be extremely tolerant, there was a huge height difference in uninoculated plants. So if you look at the very back of the left picture, you can see the tallest one, it's head over the whole row. That one has no nematodes, and that's the reason. Same with the picture on the right, you can see inoculated versus uninoculated. So regardless of tolerance, it still is important to keep your nematode populations in check if you want to get the highest, most optimal yield possible. But you can see overall the plants were very healthy and were taken great care of by the crew out at Waikea. Thank you very much for helping us with this experiment. So when we took these quarterly nematode extractions, we would just do subsamples from random plants in each box and compile those and extract the nematodes using a Berryman funnel. And this is just an average per 10 gram of roots, how many nematodes we got out. And this is averaged over the 12-month period. So the lowest nematode population was in the fluopyram treatment, vellum 1. And the highest we saw was unfortunately in the Nimitz treatment. So this wasn't statistically significant, but you can see the trends here. Venerate and broadform were similar to the untreated inoculated water control. And looking at the burrowing nematodes in cinder samples, where we would randomly collect 100 cc's of cinder in each box. Nimitz actually was the lowest, and I have a feeling that probably Nimitz is effective against burrowing nematodes, but maybe more so within the soil system and in the cinder media instead of within the plant itself. And that's why we were seeing more success in the cinder beds. However, vellum 1 was also equally effective at reducing the nematodes in the cinder. And Venerate was no different than the water control. So we decided we would do a root health rating since we could pull the plants all the way up. So this is just some preliminary results. We're just getting through the plant material now. But we rated the roots on a scale from 1 to 5, 1 being the picture on the left, white, healthy roots, which is what we all want to see in our farm, compared to the picture on the right where you see quite a bit of rot. That picture on the right probably isn't a 5, it could be more like a 4. Usually a 5 would just be a rotten gobo with no roots. But just to give you an example of the extremes for the rating scale, that photo is actually from one of the Venerate treated boxes. So unfortunately, we didn't see much success with that treatment. The picture on the left is uninoculated, no nematodes, just healthy anthurium roots. So of course, uninoculated got a #1 rating. That's what we all want to see. But as far as once you put the nematodes on, Vellum 1, the fluopyram only treatment, was the highest and most healthy root system, followed by Broadform and Venerate, and then Nimitz was equal to the untreated water control. So we still have a lot more data analysis from that experiment to go. So next year, I'll give you an update on what the yield results were, what the root weights were, and we'll just keep working on that and get that information to you later. And so the next project I would like to talk about is the burrowing nematode transcriptome and new genes for host plant resistance. This project is headed by Paulo Vieira, who works at the USDA ARS US National Arboretum in Virginia. And we're very grateful to have him on board. He's a molecular biologist and a nematologist and has a lot of interest in this field. So I'm just going to share with you some of the work that he's been up to. And last year I mentioned that we did the burrowing nematode transcriptome using an Illumina mRNA sequencing analysis from a mixed population of burrowing nematode collected directly from an Anthurium farm. And we obtained 106 million paired end reads. And the bottom right photo is just showing the comparison of the transcripts that we saw with other commercially important plant parasitic nematodes. So the first thing when you get all this information is you just kind of want to classify these transcripts into different sections so you know what their function is. But you know, there's a lot of information there, and this could be years and years of work. So you have to narrow it down. So the area Paulo decided to look into was investigating effector proteins. And effector proteins are what allow the nematode to parasitize the plant. So by disrupting this, you can disrupt the whole cycle of nematode parasitism. So he started with a reference transcriptome, and he assembled 44,478 transcripts. And then he narrowed that down and like, okay, let's just do a gland cell transcriptome. So he micro-aspirated the gland cells and then focused on the secreted proteins that he got from there. And he found 540 putative secreted proteins. So he's still busy. He has a lot of work to do still. But he's come a long way and made a lot of interesting observations. So what he's doing right now is data mining the parasitism-related genes, and mostly started with focusing on cell wall degrading enzymes, which is critical for how the nematode feeds on the plant system itself. And if you take a look at this picture of the nematode, you can see the glands on the right, and they just play a very important role in allowing the nematode to feed. So he also discovered some other parasitism genes that were responsible for modulation and protection from host defenses, and then some potential candidate effector genes with unknown function that no one had discovered yet or worked with. So that's very exciting for the future in researching that. So he took these genes, these pioneer genes that are new to everyone, and also the ones that had transcripts with similarities to other nematode parasitism genes that were located within the esophageal glands. And then he did in situ hybridization, just to make sure that he could see expression and localization of the candidate effector genes. And he did. He had very nice results from that. And with those, and also with the pioneer genes, he's done a number of different genes and confirmed them, also with a molecular analysis. So what does that mean for the Anthurium industry? Well, these genes could be very important to developing host plant resistance in Anthurium. But first we have to start at the beginning. So he developed double-stranded RNA constructs for us, which would silence these effector genes and also some movement genes that we can test and transform them into carrot hairy roots. He And then we're screening the transformed roots in a burrowing nematode resistance assay in the laboratory. And then when we can narrow it down to see what genes have the most potential, these genes will be successfully transformed into Anthurium. And this is a photo of what the roots look like in the lab. And it's a very good system for evaluating quickly a number of genes against this nematode. And here's a picture of the hairy root plates with the burrowing nematodes coming out. So just to wrap up, I wanted to give you a reminder of the first part of my talk. Remember the IPM plan for managing burrowing nematodes, and that is prevention, start clean, stay clean, watch water and worker movement, eradication, hot water bath to disinfest cuttings, steam sterilization of cinder, host resistance, plants with resistance or tolerance to nematodes, which we're working on right now, and curative control using fluopyrium applications. So thank you to all my collaborators, grower collaborators and UH collaborators, and thank you especially to Kathy Mello, who works with me in the nematology lab at PBARC.
Thank you, Dr. Myers. It looks like you got a lot of work. We look forward to your future results.